Hello, everyone. My name is Nicholas Piccolo, and my lecture today will be on the ecotoxicology of nanomaterials. So in this lecture, we'll be focusing on the following topics. We'll be focusing on the history and manufacturing of nanotechnology and nanoscience. We'll be focusing on how to classify and nanoparticle classification, nanoparticle reactivity, the formation and behavior of different nanoparticles, nanoparticle toxicity and how nanoparticles contaminate a body, as well as nanoparticle research and remedies. So the history of nanotechnology and nanoscience. So to get into context, what defines nanotechnology and nanoscience? So nanoscience is the study of structures and molecules on the scales of nanometers between 1 and 100 nanometers. And nanotechnology is the technology that uses nanoscience in practical applications and concerned with the use of structures by controlling their size, shape, and properties exclusively on the nanometer scale, as well as has to take advantage of certain systems on the nanometer scale. Neuroscience and nanotechnology can trace their roots as far back as the ancient Greeks of the 5th century BC, who questioned how far matter could be subdivided. Specifically, the Greek philosopher Democritus, who was curious as to whether or not matter could be infinitely divided, or if there was a limit to how far matter could be divided into smaller portions, and whether or not very small pieces of matter had any practical use whatsoever. In 1959, physicist and Nobel Prize laureate Richard Feynman proposed the application of what would be at the time theoretical nanotechnology at a Caltech lecture dubbed "There's Plenty of Room at the Bottom" in Caltech. During this lecture, he hypothesized that it would be possible to conduct mechanical activities on the nanoscale. Examples he used being writing all 24 volumes of the Encyclopedia Britannica on the head of a pin, or creating robots that could also create additional robots on the nanoscale, creating nanoscale robots that could self-replicate. It wouldn't be until 15 years later that Norio Taniguchi, a Japanese physicist and scientist, officially dubbed the term for nanotechnology, that being the processing of separation and consolidation and reformation of materials by one atom or one molecule. After he officially dubbed the term nanotechnology, there became a increase in practical applications for nanotechnology. There was over time, there was many breakthroughs in the field of nanoscience and later nanotechnologies, such as the development of specialized microscopes, um, such as the scanning tunneling microscope at IBM's Zurich research laboratory by Gerd Binning and Heinrich Röhner, as well as other breakthroughs such as other precise microscopes used in the study and manufacturing of nanomaterials. It was also at this time that two new nanotechnology manufacturing techniques were developed, the top-down and bottom-up approach when it comes to manufacturing nanomaterials, as well as a couple years later, the discovery of specialized nanostructures such as buckyballs, those being the products of specialized carbon bonds on the nanoscale, which would be later on responsible for a variety of both nanoscaled carbon structures such as carbon fibers, polymers, and um, nanotubes, as well as for the formation of the first metal-based nanoparticles. So now we're going to go into nanotechnology manufacturing. So as mentioned before, we have the bottom-up and top-down approach. So the top-down approach involves the creation of both processes involve the creation of nanomaterials, but um, from different materials, different starting materials essentially. So the top-down and bottom, sorry, the top-down approach of manufacturing nanomaterials involves taking a bulk material, that's just say a large conglomerated mass of materials, and breaking it down into particles, specifically via precision engineering and lithography. And excuse me, the bottom-up approach involves taking very small materials such as atoms and electrically or chemically, physically manipulating them such that they form larger bulk materials or larger clusters and eventually form nanoparticles. Um, the um, the ultimate goal of both manufacturing processes, even though they require different processes and develop different techniques, ultimately creates nanoparticles as a result. Nanoparticle classification. Okay, so what defines a nanoparticle? Nanoparticles and nanomaterials are characterized by having at least one external dimension that is less than 100 nanometers long. That's for reference, 1 millionth of a millimeter. For context, when most nanoscale materials are too small to be seen with even conventional microscopes. Again, a specialized microscope is required to properly study and manipulate nanomaterials. Nanomaterials can come in the form of particles, tubes, rods, or even fibers, but can also take the form of cubes, spheres, tetrahedron, and rods, especially if they've been engineered or manufactured for a specific purpose. Nanomaterials are not exclusively synthetic. They can be either naturally occurring, such as mycelius, liposomes, and layered biopolymers, intentionally created, such as engineered nanomaterials, or exist as a byproduct of some industrial purpose, such as the manufacturing of copper or gold manufacturing processes which produce nanomaterials as a byproduct. So to go more into depth about specifically designed nanomaterials, like engineered nanomaterials, engineered nanomaterials are unique in that they have unique magnetic, electrical, and optical properties that were especially tailored for a specific purpose. So engineered nanomaterials can include pharmaceutical products such as products that target specific cells or even large-scale organs, products that can reinforce or reduce alter some kind of property of an existing material, such as cement or even fabric. And something that's used more and more often these days, environmental solutions and environmental remedy tools that can bind with or neutralize toxins. So one example of this would consist of activated carbon, which is a charcoal-derived carbon matrix that can be used to capture and isolate PFAS chemicals, also known as forever chemicals, by forming covalent bonds that are strong enough to essentially bind with the PFAS and allow for its safe extraction. This is in fact the only way to remove PFAS from an environment, given the resilience and durability of PFAS chemicals when it comes to various degradation processes. PFAS is a very resistant chemical overall, and so far the only solution we found to neutralizing it and extracting it involves essentially binding it to activating carbon. Despite the deliberate nature of the specific nanomaterials, as well as certain other nanomaterials, um, nanoscience is still a very relatively new field, and even in the case of engineered nanomaterials, which were designed for a specific purpose, they have a very unknown effect when it comes to their potential health effects on both ecological systems and biological organisms. So as you can see here, there are different nanoparticle forms and like configurations. We have a couple squares, we have a couple of hexagonal nanoparticle forms. One thing to note is that nanoparticles, as you can see here, will react differently based on a variety of factors, but mainly those being their surface, their first surface shape, excuse me, as well as their electrical charge. Nanomaterials with a similar shape will usually behave differently than depending on what kind of environmental factors or environmental stimulus play a role in their environment. So nanoparticle reactivity. So in terms of nanoparticle reactivity, a greater emphasis is placed on the surface material surrounding the nanoparticle rather than the core nanoparticle itself. That's to say that nanoparticles react largely is derived largely from both the nanoparticle, but also like the environmental factors. What the nanoparticle encounters, like comes across, will play a massive part in its reactivity. This is because nanoparticles, when they enter a heterogeneous environment, especially a fluid or specifically a fluid like a liquid or a gas, will cause smaller structures such as atoms, molecules, and macromolecules, for example, to cluster around and attach themselves to the surface of the particle and bind either strongly or weakly. Particles with the same material can show completely different behaviors, largely due to differences in electrical charge, size, and even surface coating of the nanoparticle. The organic or inorganic matter that comes into contact with the nanoparticle also plays into what kind of reaction and what kind of toxicity the nanoparticle has, like the properties of the nanoparticle. In general, if a large mass of nanoparticles which have bulked off together starts to, which have bulked together, like aggregate together, starts to break off, their surface chemistry changes and becomes more chemically reactive. This has a lot to do with the concept of surface to volume ratio, which largely determines the reactivity of nanoparticles. So that's to say that the higher the surface volume ratio, the higher or the more likely the nanoparticle is likely to react. Nanoparticles with a relatively smaller size have a higher surface to volume ratio. You can say, for example, like a nanoparticle that has more surface area will essentially have a higher chance of reactivity than a larger nanoparticle. Um, Nanoparticle toxicity effects are also the byproduct of oxidative stress, although this is very common with metal-based nanoparticles. Various different nanoparticles are believed to have different causes for toxicity. So specifically, what is oxidative stress? Oxidative stress is due to an imbalance between the pro-oxidants and antioxidants within a biological system. So, excuse me. Pro-oxidants oftentimes includes free radicals or chemicals with unpaired electrons, which makes them highly reactive and eager to bond with other chemicals in order to stabilize themselves. This also includes various different reactive oxygen species, such as hydrogen peroxide. Antioxidants are required, in essence, to quickly react with the toxins, with the highly reactive pro-oxidant molecules in order to stabilize them and not let them essentially go amok. So if there's an increase in pro-oxidants, the body will oftentimes try to trigger a increase in antioxidants in order to balance out the levels of pro-oxidants and antioxidants. Um, Common examples of antioxidants oftentimes include various enzymes and certain vitamins such as vitamin C and vitamin D. Um, what happens with nanoparticles, or oftentimes not nanoparticles, will simulate the behavior of peroxidants and essentially kind of tip the scales in favor for the peroxidants, which again, disturbs the balance between peroxidants and antioxidants and triggers essentially oxidative stress, which comes with a number of various health effects. So, the imbalance of between antioxidants and peroxidants will oftentimes result in a number of health side effects, oftentimes such as damage to cells, damage to protein structures and DNA, as well as increased cellular aging and the disruptions to the regulation of various cellular activities such as cell growth, defense, and reproduction. Nanoparticles. So now let's talk about nanoparticle formations and behaviors. Nanoparticles will behave in different ways depending on the material of the nanoparticle comes in contact with. So nanoparticles can come into contact with three different types of materials, those being organic, inorganic materials, and other nanoparticles. And they can essentially form aggregations or coronas. When a nanoparticle comes in contact with different types of material, or organic or inorganic, or even other nanoparticles, it will experience a change in its electrical currents or electrical field, as well as certain other changes to its properties, which will alter the toxicity level for the nanoparticle. So let's begin discussing what are the different nanoparticle formations, and let's start with aggregations. So aggregations are essentially excuse me. Aggregations are essentially what happens when a nanoparticle comes in contact with other nanoparticles. When a nanoparticle comes into contact with other nanoparticles to form large irregular clusters of nanoparticles. These clusters or aggregations are relatively hard to break due to the high number of surface connections between each nanoparticle, um, and essentially do not break apart even if enough kinetic force has been produced. Breaking down these nanomaterials, however, is possible through the use of intense heat, but this is oftentimes, even though this means of breaking down these aggregations is somewhat cost effective, this results in essentially the saturation of nanoparticles into the atmosphere, which is counterintuitive. The other form we have to talk about is the corona, which is essentially nanoparticle and biological material, which happens when biological material and nanoparticles come into contact with each other. This will cause bonds between organic and biological materials, such as proteins or polymers, which will form a what's called a corona around the nanoparticle. Now, a corona is essentially a organized kind of shield made up entirely of organic or biological material that surrounds the nanoparticle. And the different layers for this corona depend largely on the strength of the bonds between the proteins and the nanoparticle itself. Oftentimes, when testing for toxicological effects, toxicologists will oftentimes determine the effects of the nanoparticle based largely on the corona itself. So as you see here, we have some examples of different particulate formations. Left side, we have essentially a nanoparticle, nanoparticle agglomeration. And on the right, we have a nanoparticle corona. As you can see here, the nanoparticle agglomeration is very irregular in terms of its shape. And you can tell there's like a lot of relatively strong bonds between the nanoparticles. And the nanoparticle corona is somewhat smaller. These are several nanoparticles, whereas this is only one nanoparticle. But there's a very ordered, structured organic sheath around the nanoparticle, that being the corona. To go more into depth, so let's focus on the agglomeration nanoparticle structure. Here we have essentially one lone nanoparticle that's over time comes in contact with other nanoparticles to form what's essentially a agglomeration. And as you can see, this agglomeration essentially is somewhat irregular in terms of its shape. It has a relatively strong connection, a relatively strong bond, which makes each particle bonded to the regular mass in a very strong way. And it tends to be of a relatively large size due to the fact that this is several nanoparticles that have come together. In this case, now we're going to take a look at a corona nanoparticle formation. Here we have another lone nanoparticle that has, over time, come into contact with organic matter to form an organic corona. This organic corona is definitely smaller than the other nanoparticle formation, but it is far more organized, and you can say it's far more structurally sound. It's not this chaotic jumble of nanoparticles. It's actually a very very rigid structure. Something else to take into account is the fact that corona nanoparticle structures or formations are actually smaller than agglomerations of nanoparticles. This actually increases their toxicity, or one could say lethality, because these nanoparticles can infiltrate the body a lot easier and damage or bypass cells far more easier than agglomerated nanoparticles. Nanoparticle toxicity. So how do nanoparticles contaminate biological systems? Um, this is actually a field of this is actually a topic in toxicology which is really well known. Nanoparticles will contaminate a body either as a surface contaminant, an agglomeration, or a protein corona. Those are the three forms that excuse me, nanoparticles will contaminate a biological system. Nanoparticles can enter the lungs, the gut, or the skin. The most common form of nanoparticle contamination is by inhalation, but there has been an increasingly higher number of nanoparticle contamination via indigestion due to nanoparticles becoming more and more used in the field of being used in as food products or food supplements. Just take a drink there. So nanoparticles, once they enter our body, will contaminate every part of the body. That includes the intestinal tract, the excuse me, so say the body cavity, as well as even the brain. How this works is because our natural bodies have a natural defense mechanism against particles that take in the form of that defense mechanism against particles being what's essentially mucus. However, nanoparticles are small enough to completely bypass this mucus defense system and essentially enter the body without coming into any will not be stopped or collected by essentially mucus. So in the case for nanoparticle contamination in the brain, what will happen is that nanoparticles will be um, inhaled through the nose and completely bypass the olfactory bulb located between the nose and the brain, and essentially find their way into both the brain and the central nervous system, causing a number of disruptive or neurodegenerative effects. Now, nanoparticles are the biggest danger from nanoparticles is that they cause damage to cellular activity. Nanoparticles are oftentimes too small to cause damage to organs, and so when they enter a body, they'll oftentimes disrupt cellular activity by infiltrating the cells themselves and cause damage to different organelles within the cell. So common forms of nanoparticle damage to cellular activity often takes the form of physical damage to the cell via lysosome penetration. Lysosomes are organelles that are responsible for digesting or recycling cellular material via special digestive enzymes. Lipid peroxidation and production of reactive oxygen species via vesicle infiltration. Vesicles are essentially a means by which the cell membrane can fold to allow matter to enter or exit the cell. Physical damage to the via disruption to the mitochondria. The mitochondria is an organelle responsible for respiration and energy production within the cell. This is especially troubling because the mitochondria has a double membrane, and yet nanoparticles have been shown to be able to completely bypass this double membrane sheath around the mitochondria. Um, Exposure to toxic ions via membrane damage. So nanoparticles can damage the membrane, which exposes cells to um, different toxic effects. One common problem is that nanoparticles can cause protein misfolding via damage to the Golgi apparatus, that being organelles responsible for transporting, managing, and changing proteins and lipids within a cell. And finally, DNA damage via infiltration of the nuclear envelope of a cell. So nanoparticles can penetrate the nuclear envelope to essentially damage the DNA within the cell. Nanoparticles can also disrupt different bodily functions such as macromolecules, hormones, different proteins, and even enzymes by essentially either inhibiting or even behaving similar to these molecules. Nanoparticles have shown the ability to essentially interfere with cell signaling processes and simulate how chaperones operate and manage how proteins fall. So essentially, nanoparticles will disrupt how chaperones behave to cause proteins to misfold, which can essentially cause a number of problems such as the formation of amyloid-like biostructures, which bring similar a bunch of neurodegenerative diseases to a living organism. So now we're going to talk about nanotoxicology research and remedies. These are the research and solutions we have when it comes to treating nanoparticle contamination. So it's important for us to understand how nanoparticles behave once within a body, and this will require a variety of new technologies and research or even predictive models that that are still in testing or currently being researched on. Um, so the most important data sets when it comes to nanotoxicological research and remedies has to do with physicochemical traits, excuse me, as well as metabolic profiling and gene expression. So physicochemical traits are the have to do with the physical and chemical attributes of nanoparticles. Those being the three main traits that define nanoparticle behavior: the size, the shape, and the electrical charge of nanomaterials. This can be very difficult given the small size of nanoparticles and difficult when it comes to observing them in a controlled environment. Gene expression and metabolic profiling will also be required in order to assess how not only nanoparticles infiltrate cellular activities or imitate macromolecule behavior, but also as to how cells will change their expression or their behavior once they've been contaminated with nanoparticles. So let's begin with omic-based clinical discoveries. Omic-based clinical discoveries are as a field of medicine that uses high throughput technologies and techniques such as sophisticated robots, data processing and control software, liquid handling devices, and sensitive detectors to measure molecular makeup or biological samples, specifically tissues or cells, for signs of disorders or mutations. This is essentially the use of automated equipment to test thousands to millions of samples for biological activity at the cellular or molecular level. Um, Omic-based clinical techniques have already been used in the field. One example would include fluorescent polarization, excuse me, which is a rapid and quantitative analysis of diverse molecular interactions and enzyme activities via a light source that passes through a polarizer. There's also affinity purification, which is the separation of molecules in a solution based on differences in binding interactions with a ligand that is immobilized to a stationary material in its solid phase. So omic-based clinical techniques are generally used to develop new diagnostic tests and involve fields of transcriptomics, which is to say RNA and DNA relations, genomics, proteinomics, study of proteins, and macrobiomics, which is the study of microbiomes. It's focused as to how disease develop and allows for the creation of new targeted therapies to identify and treat disease-prone patients. And thus it is believed that this omic-based clinical techniques can be used to develop new toxicity screening tests for nanoparticle contamination, as well as new biomarkers for potential traits for nanotoxicology. There are some downsides to omic-based clinical discoveries. Omic-based clinical discoveries provides limited data into the complex behavior of certain cellular pathways and biological activities within a cell. That's to say that omic-based discoveries, despite being very sensitive to very minuscule readings, they're not sensitive enough to determine different biological, very minuscule or very small biological activity within a cell. Omic-based clinical discoveries, however, do take into account the environmental factors which determine nanoparticle toxicity. Next, we have QSAR predictive models. So QSAR stands for, quantitative structure activity relationship modeling, which is a computational tool used in chemistry, biology, and bioengineering. It involves the application of mathematical models to predict the behavior and properties of chemical activities given their molecular structure and physical attributes. This helps to establish a relationship between the chemical structure and biological activity based on mathematical models or computation. In theory, QSAR can analyze reliable experimental data from nanoparticular structures, biological activity, and physicochemical processes of the nanomaterial itself. It's the benefits to QSAR predictive models is that it saves time, money, and lives when conducting studies. QSAR essentially is a virtual model. That's to say that no labs or technicians are required. And provided you have enough data, QSAR can organize and produce results at a faster and more reliable rate than with human researchers. QSAR is also completely virtual. That's to say that it does not require the use of animal test subjects or even human test subjects in order to determine the results of a study or a specific therapy. Now, the problem with QSAR is that the quantitative structure activity, the predictive QSAR predictive models depend largely on our knowledge and data of nanoparticles. And given how we know so little of the toxicological or physical traits of nanoparticles, as well as the fact that new nanoparticles are constantly being produced every day, means that QSAR predictive models are not a reliable way of being able to model nanoparticle behavior due to our limited knowledge and data about nanoparticles. Furthermore, nano QSAR models do not take into account the variable environmental influence that environmental conditions have on nanoparticles. Again, environmental nanoparticles largely draw their toxic effects from the environment they're in, like the different materials they come into contact with, and QSAR predictive models do not take that into account. One promising remedy that has arisen to treat nanoparticle toxicity is the pegylation process. So in medicine, pegylation involves a molecule, oftentimes a specially designed protein, to be attached to a synthetic polymer called a polyethylene glycol or PEG shield to essentially protect the molecule from being absorbed or broken down by cells or enzymes, as well as to make the molecule less recognizable by other cells or the immune system. This will be a little different for nanoparticles. So in the case of nanoparticles, this process will alter the dispersion state of the particle, which will limit its bioavailability and limit its toxic effects when it travels throughout the body. This process is due to PEG having the ability to essentially shield the nanoparticle from essentially from reacting with other other pieces of material by essentially shielding the surface charges and imparting larger circulation time to make the nanoparticle less reactive with the surrounding environment. This process can be done with other molecules other than PEG, such as polyvinyl pyrrolidone or various polymers or even polyvinyl alcohol. However, PEG is the most popular option due to its ability to essentially shield surface electrical charges. There have been 2 experiments conducted with pegylating essentially nanoparticles. In one of these experiments, platinum cobalt nanoparticles showed no signs of toxicity after being subjected to the pegylation process. This experiment took course over the course of 60 days. In another experiment, PEG-coated gold nanoparticles showed less hypotonic outcomes than uncoated gold nanoparticles within 2 populations of sporadic Dolly rats. So one population of these test rats was given nanoparticle contamination, and the other population, the experimental population, was given nanoparticle contamination along with a PEG therapy, and it was determined that the treatment population showed less signs of toxicity than the control population. This may mean that gold nanoparticles may actually be safe for biomedical application and usage in various fields of biomedicine and do not risk harming the biological systems they're treating. So over here we have 2 examples of the QSAR model and pegylation. The QSAR model does not take into account environmental factors, but it's focused largely on the studying the the particle in question or nanoparticles in great detail. And on the right, we have essentially an example of the pegylation process when used in medical applications. On this slide, we have the pegylatation process when applied to a nanoparticle. So this yellow portion is the nanoparticle core, and as you can see here, these excuse me, various chains of PUG are essentially coating the nanoparticle to render it inert. So when it comes to finding solutions to nanotoxicology, arguably the biggest obstacle has nothing to do with lack of technological breakthroughs or rampant nanotechnological usage. Uh, the biggest problem to nanotoxicology is the lack of a universal classification or definition for nanoparticles that is respected um, across different countries. So different different toxicological agencies have different definitions for what a nanoparticle is or what a toxic nanoparticle is justified as. Here in the United States, at least, nanoparticles are considered to be particles that are on scales of 1 to 100 nanometers long. However, in other toxicological agencies will oftentimes consider nanoparticles of slightly larger sizes to also be considered as nanoparticles. One example would be microplastics, which, while they can exist as in sizes of 1 to 100 nanometers, they can also exist in sizes between 1,000 nanometers to maybe 5 millimeters. And even larger microplastic sizes, such as above 1,000 nanometers, can have similar effects to conventional nanoparticles. And so some toxicological agencies would consider larger particles to be nanoparticles, and this can get in the way of creating a reliable model or reliable treatment program for nanoparticles. There's also no standardized procedure or experimental design or data reported protocol that would be needed um, to illustrate or determine what is considered as a toxicological effect from a nanoparticle. This also means that there is no basis for forming universal predictive computational models for what would be nanoparticle contamination. There have been various agencies which have been tasked with studying and managing nanoparticle contamination. So here in the US, the FDA established the NTF, or the Nanotechnology Task Force, to both regulate the manufacturing of nanoparticles as well as to head the decontamination and the study for nanotoxicology. And relatively recently, the Chinese government established the CDE, or Center for Drug Evaluation, for essentially a similar goal to both manage nanoparticle saturation and manufacturing, as well as to find new ways to treat nanoparticle contamination. However, even with these different agencies, our ability to remedy or pool our resources to essentially solve the problems of nanoparticle contamination is very limited, because all these different agencies have different definitions for what constitutes a nanoparticle or nanoparticle contamination. And as such, it's very difficult to essentially have these different agencies collaborate their data and find create essentially a unified solution when it comes to determining how to resolve nanotoxicological problems. I want to thank you for your time, and I want to thank all these researchers for all the work they put into when it comes to determining the toxicological effects of nanoparticles. Here is a source. Here's a list of my different citations used in this lecture. I want to thank you for your time and have a good day.